This is podcast number 24. All right, we've been talking about replicating DNA, forks and loops, and mutations are most likely to happen while DNA is getting replicated. So there's the error rate in the polymerase, and then there's just mistakes that happen as a result of doing this big molecular machinery sort of situation where you're trying to recreate a molecule with literally millions of parts. So mutations are common. You'll see in the next podcast that there are lots of mechanisms to fix mutations, and mutations that persist, say in the human population, are relatively rare. Maybe one new mutation shows up every 200,000 years of human existence in terms of persisting in the population. So that is considering that most of the target for a DNA mutation, most of your DNA in your body is going to be in your somatic cells, in your body cells, and that won't pass a mutation on to the next generation. Only your gonads pass it on to the next generation. And it's also considering that even though the chance that a mutation happens is frequent, it'll also frequently get fixed. So if a mutation happens, there are many mechanisms to fix those mutations, and that's the way it normally works. And rarely do you have something that gets outside the mechanism of fixing that mutation, and then also happens in the germline, in the eggs or sperm, and also gets passed on. That's what's rare. But a mutation happening is not rare. So, what kinds of mutations can happen? Well, you can have these things called tautomers that form, and tautomers are just rare alternate forms of a base. So if the predominant form of adenine like this, a tautomer might be moving one of these hydrogens over here to this nitrogen, and then switching the position of that double bond. And this would be the tautomeric form of adenine. And these tautomers form non-standard base pairs. So for example, a rare tautomer of thymine may base pair with guanine instead of adenine, for example. So this is the most common form mutation that would happen. And this non-standard base pairing can cause DNA replication error because one of these two strands is going to be used as a template to replicate the other strand, and it's going to use standard base pairing. The DNA polymerase is going to use standard base pairing to replicate the other strand. So if you have a tautomer non-standard base pair in there, that's not going to be replicated in the following strand. Another place of frequent mutation can be in highly repetitive DNA. So we talked in a previous podcast about how most of your genome is made up of this repetitive DNA, and that gets copied along with everything else that gets copied when you make a new chromosome. And that can be particularly tricky to copy because if the polymerase lets go of the strand while it's copying, the replicated strand can base pair with itself since it's just a bunch of repeats. So in here it's CAG repeats, and then you can form this hairpin loop where you have CGA, CG, GC, CG base pairing. You would also have some mismatches in there, like A with A, but still these standard base pairs, C with G, G with C, C with G, have enough hydrogen bonds to hold this hairpin loop together. And because it's just a bunch of the same sequence over and over and over again, it's still going to base pair with the original template strand. So if this sort of hairpin structure happens, and then the polymerase comes back on to replicate this strand, it just keeps going uh, where it left off, and where it thinks it left off is now shorter because it's made shorter by this hairpin. So as a result of that, this section may get even more repeats added onto it. And we also know this from if we look in repeated regions of DNA, they tend to be, the number of repeats tends to be variable even among individuals. How many repeats I may have in one of these regions versus you may have in one of these regions can be different. It's one of the things that can be used to identify individuals is how many repeats do you have in a given region. So this sort of thing happens, and it's a common mutation during replication in areas where there are lots of repeats. So again, what happens is the polymerase comes off during replication. There's this hairpin loop that forms with standard base pairing and then some non-standard base pairing, but it's stable enough. And then when the polymerase comes back on, it repeats making something that it just copied because it's now shorter because of this hairpin loop. And it keeps, and then one of the strands ends up longer as a result of it. There are these trinucleotide repeat that are associated with human disease, Fragile X syndrome, Huntington's disease, myotonic dystrophy type 1, where there are well-known repeats that are associated, that change the expression of a gene that is upstream or downstream of the repeat. So normally in Fragile X syndrome, a wild type individual would have between 6 and 50 repeats of the CGG sequence, but in Fragile X, there's between 200 and 2,000 repeats, so lots more DNA in that area, and that can affect the expression of genes that are involved, in this case, in development. Okay, another kind of mutation that can happen spontaneously is depurination or deamination. These are just hydrolysis reactions of DNA interacting with its water environment. This can happen spontaneously. So through a hydrolysis reaction, you can just get the guanine base being taken off of a nucleic acid, ending up with a missing base. In deamination, you can have this NH2 group removed as a result of hydrolysis and replaced with oxygen, double bond oxygen. So it changes the base from being cytosine to being uracil as a result of this. So in depurination, you just end up with a missing guanine, but in deamination, you change what the base is. In addition to those mutations, you can have environmental exposure to specific toxins that interact with DNA. Aflatoxin B, which is from a mold that grows on grains like peanuts and corn, can bind to DNA. It's a very potent toxin. Binds to guanine residues in DNA and therefore would modify base pairing because you've got this big group hanging off the guanine. You have also things like proflavin that binds within the structure of the DNA. It's called an intercalating agent. Binds between the two strands in the double helix and affects base pairing. Some of these are used as DNA stains. So ethidium bromide, if you've ever used that stain in a undergrad lab or doing research in order to look at DNA on, say, an agarose gel, the way it works is it intercalates into the DNA and is fluorescent when that happens, and so that's how it labels DNA. You should always wear gloves when you are working with these stains because it can do the same thing to your DNA and mutate your DNA as a result of that. And then there are these natural parts of your DNA that can replicate themselves and insert themselves someplace else in your DNA. And they're called transposable elements or transposons. This is what Barbara McClintock won her Nobel Prize for. She did most of the work in the 40s, and that wasn't really recognized until other people discovered the same phenomenon and went back to find out, has anybody done this before, and then found out that she had done most of the work in the 40s, and then she did get a Nobel Prize for it. So these are also called jumping genes. So the transposon is in red in this diagram, and what happens is the transposon replicates itself and then puts the copy of itself into a new place in the genome. So you can have mutations caused by the transposon leaving its original site, and you can also have mutations caused by where it gets inserted. So if it cuts and inserts itself into some part of your DNA that's really important, for example, for regulation of how often you make insulin or something like that. When you express insulin, that can change how you express insulin because you have now this chunk of DNA that's put into the middle of that piece of your regulatory region. Same with when things are cut out. So when we get more into the regulation of transcription, you're going to see that there are many, many, many proteins that bind to the DNA that essentially are telling the DNA, quote unquote, telling the DNA, make a transcript now or don't make a transcript now. And those proteins that bind to your DNA are binding to DNA according to its shape. And if you add a new chunk of DNA to where one didn't exist before, it changes its shape and therefore changes the ability of these proteins to bind and initiate transcription or shut down transcription. And then you can have exposure to radiation that can affect your DNA. It can be not necessarily like radiation like the Hulk got radiation, but just radiation in terms of anything on the electromagnetic spectrum. So ultraviolet radiation exposure to the sun can cause thymine dimers, so a covalent bond between adjacent thymine nucleotides in a DNA strand. They become covalently bonded to each other, and it changes how they interact with the opposite strand. So these no longer base pair after forming thymine dimers.